Okay, there we go. All right. Okay, so Tuesday's lecture, which was a recorded lecture, started off with the microscopic anatomy and differences between skeletal muscle cells and cardiac muscle cells. This table is going to contain the most of what you need to remember about skeletal muscle versus cardiac muscle. Skeletal muscle is multinucleated. It does not contain gap junctions. It is going to use individual motor units. So if you remember from 1141, motor units were recruited by size, small, medium, large. Every muscle fiber within a motor unit would contract together, but you would alternate motor units to resist fatigue. Skeletal muscle uses motor units, has an elaborate sarcoplasmic reticular system with T-tubules. Calcium comes only from the sarcoplasmic reticulum in skeletal muscle. It does not have pacemaker cells. It can have tetanic contractions, and it relies on both aerobic and anaerobic metabolic methods for energy production. For cardiac muscle, these are much smaller cells. Okay, so skeletal muscle fibers could be up to 30 centimeters in length. These cells are going to be much smaller, so they are only going to have 1 or 2 nuclei. They are connected by gap junctions. These gap junctions allow ions to flow and depolarize adjoining cells. These gap junctions allow each chamber to contract as a functional syncytium. That cardiac skeleton is going to prevent depolarization between chambers. Okay, so these gap junctions connect each chamber, so it connects the right atria or the left atria, the right ventricle or the left ventricle, so that we have contraction as a functional syncytium, a singular unit. The T-tubule system is much less elaborate, and part of that is because cardiac muscle is going to rely not only on the sarcoplasmic reticulum for calcium, but also on the extracellular fluid. And the ability to have calcium channels and get calcium from that extracellular fluid is one of the reasons why we maintain such a tiny, narrow range for blood calcium levels, so that 9 to 11 mg per deciliter. So we keep that very tight because it can affect the functioning of the heart. Calcium still binds to troponin and functions via the sliding filament model. Pacemaker cells will make up about 10% of the cardiac cell population. So we have contractile cells and pacemaker cells. Tetanic contractions are not possible. I'll go over why that occurs, and metabolism is aerobic only. So this chart is very important. The other thing to keep in mind when looking at cardiac versus skeletal muscle similarities and differences is the action potential shape. This action potential is from a skeletal muscle. It starts with a flat, stable, resting membrane potential here. Okay, so this baseline is flat. Then it has rapid depolarization due to voltage-gated sodium channels, rapid repolarization due to voltage-gated potassium channels. We hyperpolarize and come back to baseline. So this action potential represents skeletal muscle. As I mentioned, there are 2 populations of cardiac cells. There are pacemaker cells and contractile cells. Contractile cells make up about 90%, and this graph on the left represents an action potential from a contractile cell. So contractile cells also start with this flat baseline with a rapid depolarization. So far we're looking about the same, right? So that rapid depolarization is due to sodium. Okay, this is sodium regulated. But then we do not get a rapid repolarization. Instead, we have a very prolonged plateau. If you look at a skeletal muscle action potential, it's done in 3 seconds, 3 milliseconds. So you can have action potential after action potential very, very rapidly, and they can summate into a tetanic contraction. In cardiac contractile cells, there are slow calcium channels. So this plateau is maintained by these slow influx of calcium ions. Okay, so we're going to continue to have an influx of positive ions into the cell, maintaining this positive action potential plateau. It can last up to 300 milliseconds, and this is going to make sure that these contractile cells, that the ventricles have time to completely contract, the atria have time to completely contract before that next series of action potentials occurs. It also prevents tetanic contraction. There is no way to sum these up because we're already at this plateau. And so it has a very long, what's called absolute refractory period. So it cannot be stimulated with another depolarizing event because we're already so high up here at this plateau. So a very long absolute refractory period prevents tetanic contractions. And this plateau is due to slow calcium channels. Then we have repolarization with voltage-gated potassium channels. So repolarization is usually under the realm of potassium. Fast-acting sodium channels are going to depolarize. Slow calcium is going to maintain that plateau. And then voltage-gated potassium will repolarize that cell. As I mentioned, we have two populations of cardiac cells. Contractile cells, which are in this pretty action potential with the lung plateau, and then pacemaker cells. So this is an action potential of a pacemaker cell. The key feature of a pacemaker cell is this pacemaker potential. In skeletal muscle and cardiac contractile cells, they start off with a flat baseline. The pacemaker potential is going to be due to slow sodium channels. So these slow sodium channels allow a slow leak of positive sodium ions into the cell. And so we get this slant. This is a depolarization. So we have a rapid depolarization, a rapid repolarization, but it's this first phase, which is destabilizing the resting membrane potential that causes this to spontaneously depolarize and create that initial phase for the conduction system. So this slant during the first phase is called the pacemaker potential. It's due to slow sodium channels. This rapid depolarization is caused by calcium channels in addition to the sodium channels. And then repolarization is again under control of potassium. So slow sodium channels establish this slanting baseline. And the steepness of the slant will determine at what rate they're going to depolarize, because some cells depolarize like the SA node more quickly than cells in the AV node. So that is controlled by the slope of this line. But all of the pacemaker cells will start off with this sloping resting membrane potential. This is threshold. Once it reaches threshold, we get rapid depolarization by the addition of calcium and then rapid repolarization. So do you know the differences in the appearance of those action potentials, which action potential belongs to which cell population, as well as the role of calcium and slow sodium ion channels in the pacemaker and slow calcium channels in the contractile cells. Then there is the sequence of excitation. You do need to know the sequence. This is also covered in lab, and it's really pretty straightforward. It starts at the SA node. There is going to be a slight delay at the AV bundle. This is because the fiber diameter gets smaller, causing a little bit of a delay. That delay is going to make sure that the atria have time to contract before that conduction system gets all the way down to the ventricles. So from the AV node, we have, this is, sometimes it's called the atrioventricular bundle, sometimes it's called the bundle of His. To the right and the left bundle branches, which then further branch into the Purkinje fibers. So you do also need to know this diagram and the pathway, so the sequence. Modifying extrinsic innervation is going to be under control of the autonomic nervous system. At rest, heart rate is under parasympathetic control. The heart receives consistent input from both the sympathetic nervous system and the parasympathetic nervous system. But at rest, parasympathetic is going to override, it's going to have a stronger presence, it's going to have more activity than the sympathetic. So at rest, the heart is under parasympathetic control, which will decrease the rate of cardiac conduction. When we need to increase heart rate or force of contraction, then the sympathetic nervous system is going to override the parasympathetic. So fight or flight, increasing cardiac activity is the role of the sympathetic nervous system. So parasympathetic at rest, sympathetic during fight or flight situations. Okay. This is what this is showing. You don't need to know the pathway through the medulla oblongata, through the collateral trunk ganglia. That was the realm of 1141, and we'll leave it there. But do you know that at resting conditions, the parasympathetic is going to be the dominant tone? And under fight or flight, the sympathetic system will override that to increase heart rate and the force of contraction. ECG is covered in lab, so I'm not going to really go over it too much. You do need to know pretty much this graph. You need to be able to identify the wave pattern as well as the events that are associated with that pattern. So the P wave is atrial depolarization. The PR interval is going to be delay at the AV node. QRS is ventricular depolarization. You don't have to know the ST segment. The T wave is ventricular repolarization. And these are just kind of FYI, kind of interesting. This is a normal sinus rhythm. This is a junctional rhythm. There is no P wave. That means that the SA node is not stimulating the atria to contract. And a second degree heart block is when we have more P waves than QRS complexes. That conduction system is not getting all of its impulses from the SA node to the AV node. So these are just kind of interesting because they do represent how the electrical activity affects the contractile activity. So for today, we are going to cover two topics. We are going to cover the cardiac cycle, and then we're going to cover how to calculate and the factors that affect cardiac output. For the cardiac cycle, you may want to have this like written on a Post-it, that systole is equal to contraction. So something's contracting. If we have atrial systole, we have the atria contracting. If we have ventricular systole, the ventricles are contracting. For diastole, this is when the heart is at rest, so the atria are at rest or the ventricles are at rest. The cardiac cycle is the blood flow through the heart during one heartbeat. It is going to be the series of mechanical events. So we just went over, reviewed rapidly, the electrical events, and then I'll show you just. So this, we'll go over this graph a lot, but the top is the ECG tracing, and it will illustrate how the electrical events are just ahead of, this is the atria contracting. This is going to be the ventricles contracting. So the P wave, the electrical event directly precedes the mechanical event. So that's the theme of this upper portion of the graph. For the phases of the cardiac cycle, you need to understand what events are happening, what is going on with the valves. Okay, that's these circles here on this graph. And then what is going on with ventricular pressure and ventricular volume. So there are three phases to the cardiac cycle. This middle phase is often broken up into two phases. So sometimes you'll see that these are written as 4 phases. I've even seen it written as 5 phases. So the phase number isn't terribly important, but we will, for kind of clarity's sake, break it up into phases 1, 2, and 3. So all of your graphs are set up with the cardiac cycle being broken into 3 phases. Okay. In the first phase, it is called the ventricular filling phase. So the beginning of the cardiac cycle is going to start with both of the atria and the ventricles in diastole. They're at rest. So we start the cardiac cycle with passive filling of the atria and ventricles. So these are in atrial and ventricular diastole. Everything's at rest, and blood is flowing in either from the pulmonary veins or from the superior inferior vena cavas and coronary sinus on the right side. So we have ventricular filling. The first part of this phase is passive filling. The AV valves are going to open as soon as the pressure gets high enough in that atria that the blood starts, it's going to push those AV valves open. And let me go to here. So the ventricular volume is steadily increasing. The pressure is still relatively low, and this little blip here is when, at the very end of ventricular filling, the atria are going to contract and add about 20% of blood volume into the ventricles. These darker, I guess, the dark tan lines are going to indicate when valves open and close. So we are going to have the closure of the AV valves, and at the end of this is called isovolumetric contraction, while the opening of the semilunar valves. but we haven't quite gotten there. So the atria contract, the AV valves close. This is marks the end of the first ventricular phase. Kind of this. So at this point in time, at the very end of the first phase, the last event of the first phase is the establishment of end diastolic volume. This is the maximum amount of blood that the ventricle is going to have. So this is the end of the first phase. This is looking at ventricular volume. So at this point here, we have end diastolic volume. So phase 1 ends with the formation of end diastolic volume. Phase 2 is dark tan and light tan. Okay, so this is phase 2. So the ventricles are going to contract. So we have that electrical event here. It says ventricles, you need to contract. The ventricles go, okay. So ventricles start to contract and pressure starts to build, because right now, both the AV valves and the semilunar valves are closed. So this I call it 2A, is a very small part of the cardiac cycle, but it is important. It is this khaki band here. It is when we establish something called isovolumetric contraction. It's when the AV valves are closed, the semilunar valves are closed, the heart has its maximum volume, and the ventricles start to contract and start to squeeze that blood until it reaches this point right here where the semilunar valves open. So this band right here is isovolumetric contraction. The pressure builds, the semilunar valves open. Pressure continues to force blood out either through the pulmonary trunk or through the aorta. Volume shows a steady decline, okay? Because we're contracting, we're pumping blood out. The AV valves are closed, so we're not getting more blood in. Semilunar valves have opened, pumping blood out. The volume is going to steadily decline. The pressure starts to decline too, as the ventricles are going to start to relax. The semilunar valves close here, and this dicrotic notch is caused by, I'm just going to go with the left ventricle, okay? That because the events are larger in the left ventricle, but this notch is when we push blood out the aorta, it also starts to create this pressure on the backside. So this dicrotic notch is the forcing from the blood pressure from the aorta back towards the left ventricle, causing the semilunar valves to close. Okay, so semilunar valves close. This is where the AV valves are going to open. So during during these phases, all valves are closed. And the beginning of the third phase starts with end systolic volume. So before we so the semilunar valves close, no more blood is going to leave. Valves have not opened yet, so we're not getting more blood in. End systolic volume. So this is the heart with the remaining blood in it that was not ejected. So we have end diastolic volume and isovolumetric contraction occurring. End diastolic volume is the end of the first phase. Isovolumetric contraction is the start of the second phase. Ventricular ejection. The beginning of the third phase starts with end systolic volume. Then we start to get blood flowing back to the atria. The AV valves are going to open. We get ventricular filling, and the cycle is going to start again. So if we go to the whiteboard to move this, I have summarized goals 1 and 2. So the first goal of today for the new material is to identify and sequence the phases of the cardiac cycle. The first phase is ventricular filling. It's going to start with the atria and ventricles at rest. We get passive ventricular filling. At the end, the very end, the atria contract, and this establishes end diastolic volume. The AV valves are open, the semilunar valves are closed, ventricular pressure is low because it hasn't started to contract. Remember, it's still resting. And ventricular volume is steadily increasing because the phase is known as ventricular filling. For the second phase, it's broken into 2 subphases. Typo. So phase 2A and B as a whole is called ventricular systole. So the ventricles are primarily contracting during this time. That very initial part where it's kind of the dark khaki, where the AV valves have closed, but the semilunar valves have not yet opened, is called isovolumetric contraction. The split second where all the valves are closed and the ventricles have started to contract. So ventricular pressure is increasing, the volume is constant because it's isovolumetric, and the ventricular pressure increases until ventricular ejection, where the ventricular pressure is going to force the semilunar valves open. Ventricular volume steadily decreases as the blood is ejected out of the ventricles, and the pressure is hill-shaped because it builds and builds and builds until we force those semilunar valves open, and then it's going to come down as the blood is ejected from the heart. So it steadily decreases. So it's a hill shape. The third phase is isovolumetric relaxation. So at the beginning of the phase, the atria and the ventricles are both at rest. Before the AV valves open, there is a split second at the beginning where end systolic volume is established. So during end diastolic volume and end systolic volume, all the valves have to be closed. There can be no blood exiting or entering. So it's that very short moment in time where we have the maximal blood ventricular capacity versus the end systolic where it's what is left. And then the difference would be what is ejected into, for example, the aorta. The same volume should be ejected into the pulmonary trunk as well. During this phase, we have low ventricular pressure because they're repolarizing. The AV valves start to open once pressure gets high enough in the atria, and then we get that steady increase in ventricular volume as we start to repeat the cycle at ventricular filling. Okay, so ventricular filling, ventricular systole, please excuse my typo, and then which includes isovolumetric contraction and ventricular ejection. And then phase 3 is isovolumetric relaxation. So I would recommend going, read these, but then spend some time on the graphs so that you understand when the valves are closing or opening or closing or opening, where we are going to establish end diastolic volume and end systolic volume. If the heart, the ventricular pressure is increasing, and then it's going to decrease as blood leaves. And we have the pressure change and then the volume change. And this leads into the two themes of today, which is blood flow is controlled by pressure changes, and blood flows down a pressure gradient through any available opening, which is why we have to open and close the valves in that particular sequence. So blood flow is going to be controlled by pressure, and it's always going to go from high pressure to low pressure areas. The heart sounds, there are two main sounds. There's the lub and the dub. The lub is the closing of the AV valves at the beginning of ventricular systole. And then the second sound is the closing of the semilunar valves at the beginning of ventricular diastole. So close the AV valves, they snap shut due to an increase in ventricular pressure. And that occurs just before the opening of the semilunar valves, and then the back pressure from the aorta or the pulmonary trunk will snap those semilunar valves shut. And that's the beginning of ventricular diastole. So AV valves, first sound, semilunar valves, second sound. So blood flow is controlled by pressure changes. Those pressure changes also involve opening and closing the valves, and blood flows down a pressure gradient. The third goal of today is to look at cardiac output. You need to know the formula for cardiac output. So cardiac output is going to equal heart rate times stroke volume. And if you want, then the second part is we're going to look at how to affect cardiac output. So there are two components, heart rate and stroke volume. So if you want to increase cardiac output, you have to either increase heart rate and or increase stroke volume. Stroke volume is the amount of blood pumped by one ventricle with each beat. It could be the same amount, should be pumped by each. So, but it is a singular, the amount of blood pushed out of a singular ventricle. It is a rate. So cardiac output, the units are important. It's volume per unit of time. So this is usually in mils per minute or liters per minute. So cardiac output is a rate. Resting cardiac output is about 5 liters a minute. And then when we're exercising, it can go up to 20 to 25 in sedentary people or up to 35 in trained athletes. So there is quite a bit of cardiac reserve, the difference between resting cardiac output and maximal cardiac output. So if we want to increase cardiac output, we either have to increase stroke volume or increase heart rate. And we're going to go to heart rate first because it's more straightforward. Okay. As I mentioned, at rest, the parasympathetic system is in control. But during fight or flight, the sympathetic nervous system is going to secrete norepinephrine. Norepinephrine is then going to affect the receptors on the heart, allowing an increase in depolarization, and this is going to increase heart rate. Stroke volume can remain unchanged with this change in heart rate. So, but that would still affect cardiac output because cardiac output is heart rate times stroke volume. So if stroke volume is staying the same, the increase in heart rate will increase cardiac output. This chart is very helpful in keeping in mind how the factors that affect cardiac output, stroke volume, and heart rate. And if you want to increase cardiac output, you need to increase heart rate and or stroke volume. This increase in heart rate is created by an increase in sympathetic activity. This leads to this, leads to this. Okay. Now we're going to look at stroke volume. So stroke volume is calculated as end diastolic volume, so a full ventricle, minus the end systolic volume, what is left in the ventricle after it has contracted. So stroke volume is end diastolic volume minus end systolic volume. If you want to increase stroke volume, so if you want to increase stroke volume, you can either, or in conjunction with, increase end diastolic volume, so make this first number bigger, or decrease end systolic volume, make this number smaller. Okay. So if you did both, let's say we started out with 70 and systolic volume, actually, let's go with, we need something bigger. Let's go with 130 minus 40 would end up with a stroke volume of 90 mils per beat. If we increase end diastolic volume and we were to decrease end systolic volume, then you end up with a much greater stroke volume. So these can be done separately or together. There are 3 factors that affect stroke volume by affecting end diastolic volume or end systolic volume. Preload is the factor that will affect end diastolic volume. Contractility and afterload will affect end systolic volume. So we'll look at preload first. Preload is controlled by the Frank-Starling law of the heart. The farther you stretch the muscle, the more forceful the contraction. So with skeletal muscle, if you go back to the factors that affect the force of contraction, it could be the number of motor units, it could be the size of the motor units, but it's also the amount of stretch. And In skeletal muscle cells, they are at an optimal length at rest. At rest, cardiac muscle cells are much shorter than the optimal length. So they contain much more of a reserve to the number of cross bridges. So if they're short, they can only make us like 60% of their cross bridges. When they get stretched, they can make 190% of their cross bridges. So the difference between skeletal muscle and cardiac muscle is that at rest, cardiac muscles are shorter than optimal, and that allows for a reserve in the number of cross bridges that can be formed. Okay, so for preload is going to, if end diastolic volume, so the amount of blood in the ventricle increases, it's going to stretch the ventricular muscle, and that stretch in ventricular muscle increases the number of cross bridges, and the contraction becomes more forceful to eject more blood. So preload is the factor that affects end diastolic volume. You increase preload, you increase end diastolic volume, and increase cardiac output. A slower heart rate can lead to an increase in end diastolic volume, and then exercise can increase venous return. So the slower heartbeat allows for more ventricular filling time, and then exercise increases the muscular activity, which decreases the amount of blood that is contained in that venous system. So blood is more efficiently returned to the heart. Okay. But three factors that affect stroke volume, either by affecting end diastolic volume or end systolic volume, the one that affects end diastolic volume is preload, and it is based on the principle that the farther you stretch the muscle by increasing the ventricular volume, the more forceful the contraction. The next two are going to affect end systolic volume. So we want to lower end systolic volume in order to increase stroke volume and increase cardiac output. So end systolic volume is the amount of blood that remains in the ventricle following ventricular contraction. Contractility is independent of muscle length, and it is controlled by sympathetic nervous activity, which increases the number of calcium channels. So we get sympathetic activity, or norepinephrine release, sorry. It's going to use a cyclic AMP second messenger. We saw this before with adenylate cyclase. You're not going to have to remember the second messenger system. I'm just bringing it up to you to make you feel that you didn't learn it in vain. At 1141, it shows up again, but you will not have to know the enzymes that are involved. This stimulus, okay, so what we're trying to do is we're trying to decrease end systolic volume, and that is accomplished by the sympathetic system releasing norepinephrine, resulting in an increase in calcium channels. We get more calcium in, we get more cross bridges formed, we increase contractility. Positive inotropic just means that it's going to cause a stronger contraction. Negative results in a weaker contraction. And you don't need to know the specific factors under positive or negative, except for how norepinephrine and epinephrine affect the functioning by increasing calcium. Now, the third one is afterload. In a healthy individual, afterload is not going to be playing a factor. But where afterload comes into effect is in disease conditions where there's hypertension. So afterload is the pressure that must be overcome for the ventricles to eject blood. So if there is high systemic blood pressure, which we call hypertension, there is more pressure pushed against the semilunar valve. So then the ventricles have to push harder to get blood out, and more of their contractile energy is placed into forcing these valves open. And so they have less available contractile energy to eject blood from the ventricle. So afterload is back pressure, and it is going to increase systemic volume because the ventricles they don't have as much energy left to eject blood because that energy has been used to open the valves. This will reduce stroke volume and reduce cardiac output. But again, this occurs much more often in disease conditions. So to increase, if we're going to exercise and increase cardiac output, you need to increase heart rate, you need to increase stroke volume. Heart rate is under sympathetic activity. This is looking at the nervous conduction of the heart rate. So those sympathetic nerve fibers are going to run through those trunk ganglia and affect heart rate. Then we have stroke volume, which is end diastolic volume minus end systolic volume. If you want to increase stroke volume, you can either increase end diastolic volume or decrease end systolic volume. The factor that affects end diastolic volume is preload, and that is controlled by venous return. Venous return is through that increase in heart rate. It will also be affected by filling rate, so a slower heart rate, and then also muscular activity. So with preload, when you get increased stretch, so a greater volume stretching the ventricles, then that causes an increase in contraction strength. For decreasing end systolic volume, the factor that affects end systolic volume is to increase contractile strength. That is controlled primarily through norepinephrine and epinephrine, which binds to those cyclic AMP second messengers, increasing calcium, which increases cross bridges and increases contractility. Again, afterload is going to be more applicable to a disease state. We're assuming this is occurring in a healthy individual. We're not going to go over the Bainbridge reflex. At rest, parasympathetic is going to dominate, and an exercise that will be sympathetic. Chemical regulation of heart rate, we've talked about how epinephrine, norepinephrine can increase heart rate and contractility either through direct nervous stimulation or through increasing calcium channels through the second messenger system. But now what we're going to look at are the ions. The two ions that are going to be important in cardiac function are calcium and potassium. Calcium is very important because it affects that plateau, or it affects depolarization depending on the cell type. And then potassium is going to control repolarization. So imbalances in calcium and potassium are especially dangerous to the heart. Other factors that affect heart rate and thus cardiac output are age, gender, exercise, body temperature. I'm not going to go into those any further. Congestive heart failure is a disease state caused by persistent high blood pressure. So we have an increase in afterload. That increase in afterload is going to cause the heart muscle to have to exert more force. So it has to work harder. And eventually it is going to wear out. Pulmonary congestion is a left side failure, and it's going to cause blood to back up into the lungs, causing pulmonary edema. Right side failure is going to result in peripheral congestion, causing systemic edema, mainly in the legs. So it can be either from the left side, the right side, and it can cause essentially edema in either the pulmonary system or in the systemic system. The heart will try and compensate by working harder, but eventually can't continue to do that. It becomes weakened, it decompensates, and leads to the need for medical treatment. That completes chapter 17.